Good morning, students. Earlier we have studied mitosis. Now we are moving on to meiosis. Meiosis 1 is followed by meiosis 2 and followed by a formation of a haploid cell. Meiosis 1 is a reductional phase. You can say that. Meiosis 2 is similar to that of mitosis. That's why it is an equational division. Now, the other question is that interkinase fail will not occur between meiosis 1 to meiosis 2. In meiosis 1, 2n, that means the cell in meiosis 1 will be diploid cell and the content will be 4. In meiosis 2, the cell will be haploid and the content will be half of the above. That is a reductional phase. Now in haploid condition, what will happen? There is haploid means the cells are haploid and the content present over here is 1C. Now moving on to meiosis 1, there are 4 phases which is important: prophase, metaphase, anaphase and telophase 1, where prophase 1 is divided into two parts: early prophase 1 and late prophase 1, where early prophase 1 having the following event which is just similar to that of the mitosis. Nuclear membrane start disappearing, nucleolus will start disappear, centriole will move apart from each other, chromosome will start condense over here. Late prophase can be remembered by the short trick लो zoo में पापा ने डरा दिया, where L is for leptotene, zygotine, pectine, diplotene and dikinesis. And these phases are very important for the part of MCQs because maximum questions are coming from these 4 parts. And what type of question is coming? What are the event occur in these phases? Now the first phase is leptotene, where pairing of homologous chromosome is occurring. What is homologous over there? Homologous means the chromosome which are similar in size and shape. In zygotine, synaptonial complex will form because both the chromosome are joining, are very closer to each other, forming a synapsis or a junction. In pectine, crossing over or homologous recombination is taking place. Chiasma is forming over here, but this chiasma is seen in diplotene phase. So don't be confused in these two questions. Here chiasma is forming and here chiasma is seen. Independent assortment of alleles and segregation of alleles, that is occur in diplotene. Now this particular question can be asked from the genetics as well as from the cell division as well as from the evolution part of view. In dikinesis phase, terminalization will be occur because the exchange of gene sequences has been completed and now the chromosomes are at the terminal to get separate from each other. Now recombination. This recombination is initiated by a protein called REC-A protein, continued by REC-BCD protein. What recombination do? Recombination causes variation by which we all are different from each other. It leads to evolution. Next, moving on to metaphase 1. Now metaphase 1 is just similar. Now all the events will be similar as we have started earlier in mitosis. Now metaphase 1, 2 metaphoric plate will be seen because there are 2 chromosomes over there. In anaphase 1, these chromosomes move apart towards the poles. Chromosome will form over here. In telophase 1, separation of chromosome will occur. In prophase 2, same event. Metaphase 2, the question is single metaphoric plate. In metaphase 1, 2. In metaphase 2, here is single metaphoric plate will be seen. Anaphase 2, same event. Telophase 2, same event. Cytokinesis, 4 new daughter cell will be formed. Example is spermatogenesis and oogenesis. In spermatogenesis, sperms are forming and in oogenesis, ova is forming. Now let's do some question on behalf of spermatogenesis. We can see that एक cell में अगर meiosis होता है तो 4 cell का formation होता है and the 4 cell is alive. Now in a single cell, 1 mitosis occur and mature gamete will form where 2 nuclei will be formed, vegetative and germ cell. Now my question is that how many meiosis in male occur to form 100 seeds? 1 cell arise 4 cell, then how many meiosis is required to form 100 cell? Divided by 4, 25 meiosis is needed. How many mitosis to form 100 seed? 1 cell mitosis in 1 cell, 1 mitosis, 2 nuclei. So 1 cell, 1 mitosis, 2 nuclei, 100 seed, 1, that means 100 mitosis is required. If we talk about overall cell division required in a male, 25 meiosis, 100 mitosis, overall 125 cell division is required to form 100 seeds. Let's see about oogenesis. One cell, when undergoes meiosis, 4 cells will form where 3 will be disintegrate and only 1 will be alive, different from the male part. Now, the one alive cell will be do 3 mitosis to form 8 nucleus in a particular cell. How many meiosis to form 100 seed? 1 meiosis, 1 cell. Now 100 divided by 1, 100 meiosis is required. For mitosis, we can see that 1 cell completed when it continued 3 mitosis. So 100 into 3, 300 in mitosis. And overall cell division requires 100 meiosis, 300 mitosis, overall 400 mitosis is, or cell division is required. Now, moving on to amitosis. If we see amitosis, this is the process which is dissimilar to that of meiosis and mitosis, because here cytokinesis as well as karyokinesis both occurring at the same time simultaneously. Example, amoeba and bacteria. Now here is an exception that in eukaryotes, mitochondria and chloroplast both are performing a mitosis process. Now, here cell division is completed. Now, moving on to some extra points. Shape of chromosomes. This you can study in anaphase of mitosis. Now, on the basis of centromere, where it is located, we are naming different type of chromosome. If centromere is in between the chromosome, that is metacentric. Meta means in between, centric means centromere. So when centromere is in between, and if you move it in anaphase, it will be seen like this, which is called V shape. Submetacentric, L shape, acrocentric, J shape, telocentric, I shape. Where centromere always move in upward direction, you can see here P arm is getting shorter and Q arm is getting longer over here. Now what do you mean by congregation? Congregation is the term which is given to the arrangement of chromosomes onto the equator. Now here we are done with the cell division. Thank you. Next topic which I am taking is the biomolecules. So be prepared for the next topic. Thank you.